Hello? There we go. Thank you for having me. It seems that the point of this conference is to explore what is your passion. And I really identify with whoever wrote sandwiches on this board. So it's easy to say that your passion is for food when it's lunchtime. My passion is cheese. And cheese has really governed my life in some major ways. So I chose to study French in 7th grade, not because I had this lofty idea of how it was going to open up worlds for me. No, I chose French because the French teacher offered us cheese, which makes sense because France has over 300 types of cheese. It's probably the best cheese on the planet. Another way that cheese has really affected my life. So I went to Girl Scout camp several summers when I was growing up, and we had this campfire song whose sole lyric was, "It's cheese, it's cheese, it's cheese that makes the world go round, it's cheese." So I still think that cheese is important, obviously, but maybe not for the same reasons espoused by this song. In fact, I started looking at cheese under a microscope and found out that it's arguably microbes that make the world go round. There are more microbes on this planet. The biomass of microbes on this planet is equivalent to all of the weight of the plants on this planet. And in fact, you are more bacteria than human, because the bacteria in your gut alone outnumber your human body cells 10-1. So all of these seem like good reasons for trying to understand microbial dynamics. It's important to note that microorganisms rarely live on their own in isolation in a pure culture. Instead, they form really complicated ecosystems. You've learned about this in your biology classes, how ecology works. They will compete with each other for resources, or they will use one another's metabolic byproducts. And so there's a lot of microbial ecosystems that we want to explore. Unfortunately, some of these are really, really complicated. So you can't just open up a human and look at all of the millions of bacteria in their gut, billions. You can't just parse apart soil and figure out exactly what's going on. Too complicated. However, this brings us to cheese. Cheese. Cheese is a relatively simple microbial ecosystem. There's max 4 to 9 different taxa of bacteria in a given cheese. And it's reproducible. Think about the fact that cheesemakers have been making the same variety of cheese for generation after generation after generation. So cheese is a really interesting model ecosystem. And like all ecosystems, you have dynamics and change over time. So you have bacteria that come in, different populations that interact with their environment, they deacidify it, make it more hospitable for other bacteria and yeasts to come in, compete with each other. And we have some major interactions between bacteria and microorganisms that we can characterize. Namely, we have stimulatory interactions where some bacteria encourage other bacteria's growth. So circled in yellow here, we have an interaction where 2 bacteria are growing in their own little parallel lines, and where they overlap, one bacteria is encouraging the other's growth. Conversely, we can also have inhibitory interactions where one bacteria creates a radius where it's really not hospitable for another bacterium to grow. And these are just 2 examples of some really interesting interactions that occur in the human gut, occur in soil, and also occur in cheese. Most recently, I was able to move to Rennes in France, where I was able to study how cheese tastes, and I love the way cheese tastes. So it was really exciting to me to be able to figure out why that happens. While I was there, I was funded by a Fulbright grant, and this is an international educational exchange. The applications are merit-based, and they have Fulbright programs with the US and over 155 different countries that you can choose from. There are programs for teaching exchanges, and there are also programs for research scholarship. My project was to look at Propionibacterium freudenreichi, and everyone here has probably eaten one at some point, because these are the bacteria that are responsible for the holes in Swiss-type cheeses. And I was curious to see which bacteria produce different kinds of flavor compounds. This is a lot of chemical names, but what you can really recognize is the way that this influences flavor. So these are volatile aroma compounds produced through the metabolism of microorganisms that give cheese all of these different flavors. So you have cheeses that you recognize as buttery or mushroom. Yes, if you had a camembert, you can sort of detect that as well. So this should be familiar to you. And my job was to characterize some of these flavor compounds. And an interesting thing about Propionibacterium freudenreichi is that it really has a diversity of metabolic processes. It can ferment some sugars, it can catabolize amino acids, and it can also break down milk fats into free fatty acids. So this was my core of research. However, I was also exposed to research in France, working in a lab there and comparing and contrasting with labs that I've worked in the US. And I picked up some very interesting things there, including the way that STEM education works in France. It's much more concentrated and accelerated than we have in the US. So students specialize earlier, all of their classes have very practical applications, but what that discourages is independent research that you have all been exposed to starting, in high school here. And you don't have as much flexibility. So changing careers later in life, much less frequently done in France. We also have some differences in the way that we collaborate. So in my labs in the US, we would have a lot of interdisciplinary collaboration. I once worked in a lab that was half mechanical engineers and half biologists. In France, there's more of an international collaboration. They have partnerships with labs all over the world, and in addition to exchanging ideas, they also exchange people. So students, postdocs move back and forth and do these dual degrees between multiple institutions. But the biggest culture shock for me as a microbiologist is that in the US, I would go into lab on the weekends, at night, whenever my bacteria are ready to experiment on. I wasn't allowed to do that in France. In France, I worked 9 to 5, and I had lots of weekends and breaks. I had a coworker in the US who would sleep on a cot in lab when he needed to be there for experiments, and there's this workaholic atmosphere. In France, I had coffee breaks twice a day, which always involved a cake and at least 15 minutes of chatting with people. I had to go home at 3:30 on Fridays, 35-hour work week, but lest you think I'm reinforcing the stereotype as French people as never working, being lazy, they were really, really productive in between all of these breaks. And that is something that their quality of life was a little more sociable and a lot more fun in the lab. So taking all of this and synthesizing it down, I developed some useful models. I know that I'm not going to study cheese for the rest of my life. But I like the idea of using cheese as a model for more complicated microbial ecosystems that I do want to study for the rest of my life. And I know that I'm not going to live in France for the rest of my life, but I like the idea of taking coffee breaks, taking 35 hours a week to work in lab, maybe a little more. But as I go forward in my scientific career, I'd like to keep some of those work-life balance habits that I got in France. And so cheese has really affected my life. And it seems like something small and insignificant, but when you're thinking about what your passion is, it doesn't have to be something that sounds big and lofty and makes sense that it would govern your life. You can use a passion as a model for the way that you want to live the rest of your life.